For our next installment of our series on cellular respiration, we are at the end of this set of steps. So the first one, remember, was glycolysis, where the cell took in a glucose and then converted it into these two molecules of pyruvate plus some carrier molecules such as NADH, I had to think for a second there. So NADH and a couple of ATP. So that was all generated from glycolysis. And then the pyruvate are going to go into this next thing we're calling the link reaction. So the pyruvate then in this link reaction get converted into something called acetyl-CoA. And I'll go into more detail on all of this and coming up. But for now, this is an overview. So here's the acetyl-CoA, and then acetyl-CoA is going to enter the citric acid or Krebs cycle, joining with that oxaloacetate, if you remember that, going around the cycle, and then generating a couple of ATP. So we don't get many ATP out of this either, but we do get some of these molecules that we've called coenzymes, the NAD and the FAD, which are going to pick up hydrogens and electrons, and so they are the electron carrier molecules, and that all happened in this area called the mitochondrial matrix. And then now we're at that point where those carrier molecules, NADH and FADH2, the ones they've already picked up hydrogens and they've picked up electrons, are going to now move into this very final set of stages or steps, which we call oxidative phosphorylation. So in this case, we're going to get the true payoff of this molecule of glucose into many ATP, which are going to go down something called the electron transport chain, which we'll see coming up. And we're going to extract energy from these electrons that have been carried there, and we're going to use that to fuel a process that will generate more ATP. So here we go. So this is the whole process is called oxidative phosphorylation. So phosphorylation is putting phosphates on something. In this case, we want to phosphorylate ADP into ATP. The oxidative piece is what this has to do with those redox reactions. And the other piece that you might remember on this is that it's going to need oxygen. So this is the only area where oxygen is needed, and we're going to see how that works. So this pathway is going to create this kind of a chain of different carriers passing things along, and then ultimately creating this ATP through something called chemiosmosis. Now you know what osmosis is because we've seen it already, right? We know that osmosis is this movement of water, usually down a concentration gradient. But now we're going to look at something called chemiosmosis. So it's not going to have to do so much with water, although water is involved at the end, but we'll see how this works. And then this hydrogen ion concentration gradient that you're going to see here coming up. Here's the mitochondria again. We see that it has this outer membrane, and then it has this windy kind of inner membrane, and the interior of that's called the matrix. So keep that in mind. Okay. So the electron transport chain is this series of these little transporters that are embedded in that membrane on in the interior of the mitochondria. So this is you can see here that this is the intermembrane space, and this is the inner mitochondrial membrane, this whole thing in here. And within that, just like we saw before, when we see some proteins embedded in the membrane, we see these membrane proteins which are actually carriers. So this is called the electron transport chain, so the ETC. And what this is basically is that the NADH in the first one you can see here is going to give up the hydrogen ion and sends electrons along this pathway. So electrons are going to go this way. Hydrogen protons, see that H above, H plus, that just means it's just the proton of hydrogen, is going to get pumped up this direction. So we got hydrogen protons that are being pumped into this inner membrane space. We have electrons, which are getting shuttled from one of these carriers to the next, to the next, to the next. And then finally, at the end of the road, we're going to see the output over here of, we're going to get water generated. We're going to have, and that is only going to happen because oxygen is present. And so this is kind of the first piece of this complex process. But now we're using these carrier molecules, NADH, and notice on the next one here you see FADH2. And both of them are doing the same thing. They're giving up their electrons to these carrier proteins that are embedded in the membrane, and they're sending the proton part of that, the hydrogen ion proton here, up into this space, this intermembrane space. So that's kind of step one of this process. So as these are moving, I've already talked about this a little bit, we have each one of these electron transport molecules is more electronegative than the previous one, and it's going to have a greater affinity or attraction for these electrons. So that's why they keep going. So the electrons get pulled, they get attracted, and they get pulled along this whole pathway. This NADH dehydrogenase is an enzyme. You can tell because of the ASE ending. You don't need to worry about this one, but it's there. OpenStax talks about it, so this is why it's here. And each one of these are going to move along these different complexes. So this is, you see here it says the electrons from FADH2 enter the electron transport chain at complex 2. So it's basically saying right here. So first NADH comes in and it starts this going, but now FADH2 is doing the same thing. Electrons get transported from one of these to the next, moves on down the line. Protons get pumped up here. Okay, and then here's a whole bunch of different other things that are involved. So ubiquitin, sorry, there's another molecule that sounds much like this. I don't care that you know these. You don't have to know the cytochrome oxoreductase and cytochrome B and cytochrome C. All of these, don't worry about. All I really want you to know here is that there are these carrier molecules which have high affinity for electrons, and that the electrons from the NADH and the FADH2 are going to be shuttled along this pathway, and the protons, or the hydrogen ions you see here, are going to go into this intermembrane space. So that's what you need to remember from this slide. All right, next. We've got all these hydrogen ions up here. You only see one here, but there's a whole slew of them that end up being up here from all this. This is a constant process that's happening. So we have hydrogen ions up here in a high concentration, and what they are going to do through this process of chemiosmosis is that they are basically going to go down their concentration gradient, because we've created a gradient here by pumping these up into this space. And now these things are going to flow back down, and they're going to flow through this really interesting enzyme right here that's embedded here called ATP synthase. So think synthesis, think ATP, and know that ASE is that ending for enzymes, and you got it. So when the protons that were pumped up here move down through ATP synthase, this thing acts like a little bit of a machine. So it starts to spin, and part of what that does is the energy from this process is able to put a new phosphate onto ADP and form ATP. So that's a really important part of this. And why that's so important is that it's there So you can kind of see the whole thing put together. Here we have, here's ATP synthase. We're forming ATP right here. Okay, as the protons or the hydrogen ions that were up here now come down here and they're forming ATP. Meanwhile, any of those NADHs and FADH2s that came from the citric acid cycle, they are then getting moved along this electron transport chain, getting shuttled. Electrons are going this way. Protons are coming up this way into the space above. The electrons are in part moved along here because they're attracted, that ultimately they'll have an attraction to the oxygen here. And this is going to generate some water. So water is going to be one of the outcomes of that. Now, why this is such an important part of this whole process is that this is where you get the major amount of ATP. So here in this process, depending on certain circumstances, we're going to get somewhere between 30 and 36 ATP for every glucose molecule that comes into the cell. And that varies a little bit. But this is huge and very important, and not just very important, it's critical, because this is where we make the ATP, and that ATP then is used to fuel all sorts of other cellular processes. It's going to be used to do all of those endergonic reactions that we need to add ATP to move some type of pathway forward. So very, very important. So what happens if you don't have enough oxygen? Well, that's interesting. So remember that glycolysis makes, in the end, you will net 2 ATP just at the end of glycolysis. And that's going to occur no matter whether oxygen is present or not. So that's okay. But let's say you're really lacking oxygen, like you just, you're starting to, like you're running a marathon and your muscle cells are running out of oxygen. And so what we want to do is to keep generating at least some ATP. And so there's 2 ways that can happen, and both of them are called fermentation. And one of them is called lactic acid fermentation. And then in a lot of prokaryotes, they can result in alcohol fermentation. So let me show you what those look like, and we'll be kind of quick on this. So lactic acid fermentation basically says, okay, glucose came in, we went through glycolysis, and normally we'd make pyruvate, and then pyruvate would go into the mitochondria, but without oxygen, it doesn't do that. And so what happens is that instead the cell can say, okay, let's do a little detour, let's make some molecule what we call lactate. And by doing this little process of making lactate, that is going to help to regenerate some of the NAD that otherwise would not be regenerated. So we need to get back to NAD to make a little bit of ATP each time. And so this kind of bypass that gets us to making some lactate and also freeing up that NADH so that we can go back and do it again and get a couple more ATP out of this cycle is called lactic acid fermentation. And this occurs in a lot of areas. It would occur in some bacteria, so this would happen also in prokaryotes when we think about stuff that, like the bacteria that does this to make yogurt and things like that. And when we get this, if you're a runner, you can get lactic acid buildup, and that makes your muscles sore. In anaerobic species that don't really need oxygen anyway, what they do is produce alcohol. And so they basically do the same kind of bypass, but their product is going to be some alcohol. And also some alcohol. We'll leave it at that. There's a little link that you can look at in your notes or in the OpenStax. And I think I better stop there because I'm going to run out of time before I get into these last little bits, which I'll do later in class. So I may not do that in a video format. So there you go. We just did the whole thing. And we'll go back over it.